Hello everyone, welcome you all in the another video tutorial. So in this video lecture, we will discuss about an important molecular biological technique which is named as Western blotting. So let's start with the introductive part. So basically the Western blotting is an important biological technique which is also known as protein blotting or immunoblotting. So it is a rapid and very sensitive technique which is used for the detection and characterization of proteins. So let's understand first that what is the blotting. So basically the term blotting is used to transfer of biological samples from a gel to a membrane, and after that their detection on the surface of the membrane. So there are different types of blotting techniques used in the field of molecular biology, which includes Southern blotting, which is used for transferring DNA and detecting the DNA molecules. The another technique named as Northern blotting used for RNA detection, and Western blotting for protein detection. So in this video lecture, we will discuss about the Western blotting, its principle and its procedure in detail. So basically the Western blotting is a technique in which the proteins are separated according to their molecular weight with the help of the electrophoresis. So let's start with the discovery and history of the Western blotting and all the other blotting methods. So in 1975, a scientist named as Edwin Southern invented a technique which enabled analysis of DNA. So this method was named as Southern blotting over the name of its discoverer Edwin Southern. So in this method, a Southern invented a new method that enabled the analysis of DNA identity, its size and about its abundance, and this method was named as Southern blotting. So after some time in 1977, James Alvin and his co-workers, which are named as David Kemp and George Stark, who were from the Stanford University of USA, invented a technique which is similar to the Southern blotting. And this technique is created to detect RNA samples. So the scientists named this method as Northern blotting. Now in 1979, a scientist named as W. Neil Burnett invented a technique which is used to separate proteins according to their molecular weight, and he used the blotting of proteins from the SDS phase gel electrophoresis method. So Burnett gave the name of the Western blotting to this technique also according to the geographical locations of the lab of the Burnett, and also because it is in the recognition to the Southern and Northern blotting techniques. So let's understand about the principle of Western blotting. So basically the Western blot is based on the principle of immunochromatography, and this is the technique in which the proteins are separated into polyacrylamide gel according to their molecular weight. So as we have discussed that it is done under the principle of immunochromatography, so let's understand that what is the immunochromatography. So basically the word immunochromatography is made up of two words, immuno and chromatography, so which is the combination of chromatography and immunoassay. And in this assay, either the antigen for the detection of any antibody, and in that condition for detection of any antigen, the antibodies are immobilized on a nitrocellulose membrane and is discrete spots in the cartridge device, which is used for the Western blotting. So this technology is used for a wide array of tests for clinical and several veterinary and also in most of the industrial applications. Now let's discuss about the procedure and the steps for the Western blotting. So the method for the Western blotting include some steps, and there are 8 crucial steps for this method, and they are buffer preparation, sample preparation, gel electrophoresis, transfer the separate proteins on the membrane, blocking, treatment with the primary antibody and then treatment with the secondary antibody, and the final step include treatment with some specific substrate like enzymes and many other substrates. So the procedure starts with the buffer preparation, and which is followed by the sample preparation. So the sample preparation is the crucial step, and in this the cell lysate is the most common sample for Western blotting, in which the protein is extracted from the cell by chemical lysis of the cell, and this step is also called as tissue preparation. So in this step, to prevent denaturing of these proteins, some protease inhibitors are used, and the concentration of protein is determined by the method of spectroscopy, which is another most important method in the field of biomolecular biology. Now, some tracking dye named as bromothymol blue and many other tracking dyes are also added in the sample to track and to monitor the movement of those proteins. Now, the sample preparation is then followed by the process of gel electrophoresis. So the gel electrophoresis is the third step, where the sample is loaded in the SDS page, which is sodium disulfide and polyacrylamide gel electrophoresis, which is commonly known as SDS page electrophoresis. And in this, the proteins are separated on the base of their electric charge, their molecular weight, and or other some combinations of these all. So the small size of proteins move faster than larger size proteins, we know. So this principle works in the process of SDS page electrophoresis. And those proteins which are negatively charged, they move towards the positive pole, and the electric current is applied for this process. Now, in the process of gel electrophoresis, the proteins are separated according to their molecular weight, and then these are transferred to the separate proteins on the membrane. Now, the another step includes membrane blocking, and this is the most important step for the Western blotting. So in this step, the antibodies, which are also protein, they are bind to the nitrocellulose paper because of their proteinous nature. And so before adding the primary antibody to the membrane, it is non-specifically saturated or masked by using casein or bovine serum albumin, which is called as BSA. And after that, the treatment with primary antibody can be done, which is called as antibody incubation. So in this step, the first step includes treatment with primary antibody, in which the primary antibody, which is specific to the desired protein, is used so that it can form EGAB complex with these proteins. And after that, in the second step, the treatment with secondary antibody is used, in which the secondary antibody, which is enzyme labeled, is used for the antibody incubation, or the secondary antibody used against the primary antibody so that it can bind with the AGAB complex, which is made by the primary antibody. Now in the another step, the treatment with suitable substrate can be done to visualize the enzyme's action and the reaction mixture, which is incubated with the specific substrate. So basically, the Western blotting is also a quantitative test to determine the amount of protein in the sample. And in the next method, the detection of target protein can be done with different types of methods. So the methods for the detection of target protein includes colorimetric detection, radioactive detection, fluorescent detection, and many other methods. Among all of these, the fluorescent detection methods are widely used, which is used a fluorescent dye to detect the target proteins in the Western blotting method. Now the last and the final step of the Western blotting include data analysis. So now let's understand about the applications of Western blotting. So it is the most important method for the detections of proteins in the field of molecular biology, and it has a clinical significance also for the diagnosis of different types of infections. So the applications of Western blotting include detections of toxins. So those toxins which are proteinous in nature can be detected by the help of antibody-antigen interaction. And the pregnancy tests are also used. The methods of Western blotting, we can see and we know about different types of pregnancy kits which are available in the market, and these kits are also based on the principle of Western blotting. The another application for the Western blotting is diagnosis of infections caused by different organisms. They have the ability to diagnose the viral and bacterial infections in the organisms by targeting the proteins of those infectious agents. They are the most sensitive and specific test for determining size and amount of protein which are present in any material, and because of their specificity and the sensitivity, the results have very low chances of wrong results or to failure. Now the confirmatory HIV test also help a Western blot to detect anti-HIV antibody in the human serum sample, and it is also used as a definitive test for different types of diseases like Lyme disease, hepatitis B infection and many other diseases. So these are some applications or significances of the Western blotting method. So in this video lecture, we have discussed about the Western blotting, the principle of Western blotting method and the procedure of Western blotting and its significances also. Thank you.